Vai, vedete che...
So, thanks, Gigi. I switched to English because I think we have also a few English speakers in the audience because we extended the invitation to some of our network of collaborators outside of the institute itself. So, Christian Marchese is a biological oceanographer with an interest in phytoplankton dynamics and productivity in coastal open ocean. He graduated in Florence in natural sciences and then completed a PhD in environmental sciences in Quebec, in Rimouski, in Canada. And he just finished a postdoc at the University of British Columbia, University of Victoria in Canada itself. Since January, he came back to Italy, in Rome, where he started at Rome at EMBL, the European Molecular Biology Lab, where he will be working on some activity related to the Tara Ocean research voyages in Europe for the next 2 years. The work he will present now is only on the work he did in his postdoc in Canada. Hence, there are no logos of CNR, not EMBL, because all of it is past work. So I'll leave the floor to Christian to start and telling about his topic.
Yes. Okay. Thank you, Vittorio, for the nice presentation, and thank you, everyone, to be here. I also would like to say thank you to all my co-authors, Brian Hunt from UBC, Fernanda Giannini from the Federal University of Rio Grande in Brazil, Matthew Herner and Masira Costa at the University of UVic. This work was founded by MIOPAR, which is a Canadian network that promote interdisciplinary research and try to fill the gap between academia and industries in Canada. And so the presentation is organized in this way. I will start with an introduction just to contextualize the work. I will then talk a little bit about the methodology and how we organize the data, and then I will summarize the main results of this work. And finally, I will show you some preliminary results that are part of the same project. So we will not change area of interest. We will talk always about the coastal and the open ocean of British Columbia and Southeast Alaska. So let's start. Okay, the idea to divide the world oceans into homogeneous biogeographic unit, it's not new in marine ecology and oceanography. However, the number of this kind of work started to increase when it was possible to work in a simple to get satellite data very easily. This is because basically satellite data give a synoptic view of the ocean, both temporally and spatially, and also using the use of satellite data, it's relatively cheap compared to other means. So here, this figure shows the Longhurst biogeographic unit, and if we focus here, for example, on the area of interest, which is the Northeast Pacific Ocean, we can see that basically it is the intersection of three different units. Also, if we look, for example, at the Mediterranean, we see that the Mediterranean it is seen as a unique homogeneous province. So if we want to have more details, we have to work at regional scale, and so we have to do this kind of work in another with another scale to obtain more information that eventually can be useful for the area of interest. So Overall, ocean partition may provide a framework to assess environmental variability, habitat range shift, ecosystem-based approach, fishery and conservation management. Here, for example, we have an example from a practical example from a presentation at the EuroArgo user workshop. And so the figure at the top shows the nitrate concentration. So as we can see, there is a gradient from a northwest southeast gradient, and at the bottom we have the bioregionalization made by D'Ortensio and Ribera d'Alcalá in 2009. So basically the idea here is to deploy eventually float that can cover the nitrate gradient and stay in homogeneous area as long as possible. So the homogeneous areas in this case are the region. So each color here and the figure at the bottom, it's a bioregion. The infographic on the left, oh, no, better. The green box inside the infographic on the left shows the area of interest. From an oceanographic point of view, this is a very interesting and dynamic area because there are several current upwelling area, eddies. There is also a strong land-ocean interaction. It is an area that hosts several habitats for critical and important and commercially important species, and it is also subject to climate change. So in this context, two things to retain: that the seasonal phytoplankton are, because are relevant from an ecological point of view, are used to establish connection between physical change in the environment and biological productivity. Also, the identification and mapping of marine bioregion are useful in highlighting the heterogeneity of this area. For example, here, on the right, we have a biogeographic unit defined by a national science advisory process. So now this kind of process can take time, and it is based on the expert opinion, and usually the dataset that are used can be old or present some gap in time and space. So considering this, all this context and things, the goal of the work was to provide an objective marine regionalization grounded on difference in the seasonal cycle of chlorophyll A concentration that is usually used as index of phytoplankton biomass, that can be refined and continuously updated as more Sentinel-3 data becomes available. And to evaluate bioregion biological relevance in terms of variability of spring bloom onset. So now, before to show the other slide, I just would like very quickly to introduce the concept of phytoplankton seasonal cycle and phenology. So first of all, it is possible to create a spatiotemporal time series with satellite image for the same area of interest. And it is possible to work on a pixel by pixel basis. And so for each pixel here, we have our seasonal or annual cycle of chlorophyll A. And then it is also possible to characterize this seasonal cycle using some metrics. So the bloom, for example, here we have the bloom start that define the initiation of the bloom, so the substantial increase in phytoplankton abundance. We have also the bloom peak, or the bloom peak time, the time when the seasonal cycle reach its annual maximum. And then we can have the bloom end metrics and duration and so on. So I will, in the next slide, I will use, I will talk about bloom start and bloom peak times, just you to know. So this figure here shows basically the steps that we did inside a cloud computing platform to create our time series, chlorophyll A time series, that was used later as input for the bioregionalization analysis. Basically, everything here is automatized, and everything works in parallel. So the image were downloaded and stored in a first blob storage, and then 100 nodes or CPU were used to do an atmospheric correction on the initial image that were downloaded, and then stored in a second blob. And finally, again, in a parallel way, we created a weekly time series. So basically, we have each week an image of chlorophyll A. And then we used a Linux virtual machine to for the bioregionalization analysis and other kind of analysis we needed to do on the data. And this structure was, we realized this in collaboration with the computer science department at the University of UVic. And now the the Department of Fishery and Ocean Canada is working to actively working to make this workflow operational. So this is basically the time series that we get from the previous workflow, and as we can see, the spatial and temporal coverage it's quite good if we exclude the 2019 here, and there is some the percentage is not so good usually in summer, as we can see. So we used basically these 5 years to create our time series that was given as input in the self-organized map. So this figure here shows the two-step classification procedure that we used to retrieve, to delineate the bioregion. So in the box 1, we have the time series. And then in the box 2, we have the self-organized maps analysis. And okay, the self-organized maps it's a learning machine algorithm, and it's basically a black box. You can just adjust some few things basically. And one of the difficulty with this kind of learning machine is to choose the sites, because you have to organize your data in basically in an array. So if the sites it's too small, the risk is to lose information, important information that the machine is not going to see. If the sites it's too big, you can get redundant information. So we tested different sites, and then we choose the right sites with a threshold criterion on the total error. And the idea here, the work of the SOM is to explore the input data spatio-temporal pattern and synthesize the most relevant feature in a two-dimensional map. And then the output of the SOM was given in a cluster algorithm to reduce the number of classes from the initial SOM partition into an objective number of cluster or bioregion. So here we have our 10 bioregion, and overall the procedure lead to separate cluster according to the seasonal cycle of surface chlorophyll concentration. Basically, all the pixels that have a similar seasonal cycle go in a cluster. So this is basically in a simple way how everything works. So here we have the main results, the spatial distribution of the 10 bioregion, and we get 4 bioregion in the open ocean, but while bioregion 5 and 7 are further offshore, bioregion 4 and 2 are in, let's say, in a transition zone. They start on the shelf break and expand on the slope and then further in offshore water. And then we have 6 bioregion on the shelf, and for example, bioregion 1 cover the Vancouver Island shelf, bioregion 3 cover the central BC coast, and so on. Here on the right, we have this figure shows basically the timing of the bloom onset. It's based on a climatology, so it's an average. And as we can see, there are first of all, we can see a latitudinal gradient. So the blooms start earlier south of around 51 degree north of latitude, but there are also localized area. For example, here on the east coast of the Hawaii Island, where the blooms start earlier. The blooms start also earlier, for example, in the north of the Strait of Georgia, and so on. And here on the top right corner, this is basically the timing of the bloom onset for each bioregion. So the color is similar here to the map on the left. And as we can see, for example, the blooms start earlier on bioregion 1 and 2, and start later, for example, in bioregion 8, which is further north. And in the Strait of Georgia, the yellow, so the bioregion 10 start around at the end of March. Now, it is always difficult to compare this kind of results with previous studies, because you can use a different kind of time series. You can use a different kind of technique to obtain the bloom onset and other kind of metrics. For example, in this case, we used a cumulative sum, and the time at which the cumulative biomass curve reached the 15% of the total biomass was for us the bloom initiation. However, these results match quite well, very well, I would say, with previous work, especially for bioregion 10, so that cover the Strait of Georgia, which is an area quite studied. So we have much more information for this area. So let's give a look at the chlorophyll A seasonal cycle for the bioregion. So here we have the seasonal cycle of the 4 bioregions that are in the open ocean. So as we can see, they are characterized by low chlorophyll concentration. The spring bloom are slightly pronounced, and there is the fall bloom, it's more prominent, and we can see this very well with bioregion 4, so the gray one here, that also lies on the eddy, Hawaii eddy, that start to from winter to spring, and to bring nutrient from the iron and so on from the shelf to the open water. And in this area also there is iron limitation that restrict phytoplankton growth, especially large phytoplankton cell like diatoms. So here we have the seasonal cycle for the 6 bioregion on the shelf. And the situation here, it's quite different, I would say, because first of all, we have high chlorophyll concentration during all the year. And phytoplankton assemblage usually is dominated by large phytoplankton cell. And as we can see, there is the presence of unimodal and bimodal seasonal cycles. So with one peak in spring and one peak later in fall. This is a very complex system from an oceanographic point of view. So there is an area of upwelling, like the region 1, so the Vancouver Island Shelf, tidal mixing and freshwater input overall shape the dynamics of phytoplankton blooms in this area. And the only bioregion that doesn't show a clear seasonal cycle is bioregion 9, which overall it's the one close to the close to the coastline, and as we can see in cover the fjord and inlets from north to south. So here we are back to the interannual variability in spring bloom time. So we on the left here we have the maps for the spring bloom timing, and on the right here we have the same thing, but for each year the average by bioregion, and so we can first of all we can see some spatial pattern that was present in the climatology. So for example, here we can see that the bloom around close to the east coast of the Hawaii Island start earlier. It is a little more difficult to see the latitudinal gradient, and especially in 2019, for example, where the bloom a large portion of the region the bloom start earlier. And for example, in 2020, the bloom start later in a large part of the region of interest. So, and we know that these 2 years were different, with the 2020 much colder, and somehow this difference in this interannual difference and variability is captured by the bioregion. For example, here we have 7 bioregion where the bloom start earlier in 2019, and we have, for example, 4 bioregion in 2020 when the bloom start later. Basically the methodology used was able to detect, to delineate 10 bioregion for this area. This is the first biotic partition for this area, and we were able to group this 10 bioregion in 2 larger cluster. So we have the off-shell bioregion. So the bioregion are 4, 2, 5, and 7, and the neritic bioregion, the other 6. So these 2 big clusters share main characteristics, but at the same time, each bioregion is quite unique. So if you want to have if you are curious, I invite you to download and take a look at the paper that you can download from Frontiers in Marine Science. And to conclude this first part of the presentation, I will conclude this first part of the presentation with a summary. So basically, we have seen that the 300-meter resolution from the Olshi sensor allowed the delineation of bioregion boundaries with much higher precision, spanning from the strictly coastal water and inlet to the open ocean. The proposed methodology minimized subjectively and can be refined and continuously updated as more Sentinel-3 satellite data becomes available. And for this work, we used just 5 years, as we have seen, and regionalization using remotely sensed data. So this means not just chlorophyll, but eventually other kind of variables like sea surface temperature, for example, could have a practical implementation, for example, helping to optimize sampling strategy and identify target area for the deployment of observation system. And we have seen an example at the beginning in the introduction when I was talking about the Mediterranean Sea. And to conclude, in a remote sensing big data era, cloud computing platform optimized for data-intensive loads and real-time processing are needed to manage environmental monitoring effectively. So now I will show you some preliminary results. As I said, are results that come from are part of the same project, but I think that we need to work a little bit more to consolidate this results and maybe have also some other interesting information. So using now a much longer time series, we wanted to see if there are any change in environmental condition in British Columbia coastal and open ocean concerning the NPGO regime. So what is the NPGO? NPGO stands for North Pacific Gyre Oscillation. It's a climate index that was introduced in 2008 for the Northeast Pacific. For the sake of simplicity, a climate index reduces complex patterns of climate variability into a single variables. Here we can see the NPGO time series, and as we can see, there are two phases. The first one, for example, it's the orange one, it's the positive phases, and the light blue one is the negative phases. So the NPGO is an index that measures change in ocean current and circulation pattern. This means that if there are change in the current and circulation pattern in this specific area, usually they can affect key environmental variables like nutrient concentration and chlorophyll concentration, salinity, upwelling, and so on. So right now we can see that the index is negative, is in the negative phase. So this usually it's associated with reduced coastal upwelling, lower sea surface salinity, lower concentration of nutrient, and maybe more. So, but what about chlorophyll A? So here we have chlorophyll A anomaly, just for the just to simplify, but I show only this year because I want to make here things simple. And as we can see, when the NPGO is in a negative phase, it seems that there is a decrease in chlorophyll concentration in large area of the open ocean, but also along the shelf, the coast here. So the dark blue means lower chlorophyll A concentration. So there is a correlation between the NPGO and chlorophyll A anomaly. So here we are. This is a correlation matrix, and I apologize because it's not very how it is organized right now. It is not very intuitive, and but of course they are preliminary results. So I apologize for this. But the idea here was to correlate the North Pacific gyre oscillation, but we also introduced also other climate index, and we have also our parameters: chlorophyll A, mixer layer depth, and sea surface temperature. As we can see, the NPGO is the only climate index that correlate quite well with in a positive way with the chlorophyll. So this means that when the NPGO is in the negative phase, we have a reduction in lower chlorophyll A concentration, and this match with the anomaly we have seen. The NPGO is also positively correlated with the mixer layer depth. So this means that when the NPGO is in the negative phase, we have a shallow mixer layer depth. And finally, the NPGO is also correlated with the sea surface temperature, but this time in a negative way. So this means that when the NPGO is in the negative phase, we have higher value of temperature. Here we have the correlation. So because there is this difference, we were interested to eventually show these two different situations. And what we did is to create two kind of climatology, so an average. So we have a positive, we used the years when the index, the NPGO was positive to create a first climatology, and then the years when the NPGO was negative to create another climatology. So here the figure shows the first column, it's the chlorophyll, then we have the sea surface temperature and the mixer depth. The first column, the first row, it's the situation for the negative phase, and then at the bottom we have the positive phase. So overall, without to go into details, we can see that for the negative phase, we have a lower chlorophyll concentration, higher sea surface temperature, and a shallower mixer layer depth. And of course, we have about an opposite situation with the positive phase, where we have overall higher chlorophyll A concentration, lower sea surface temperature, and deeper mixed layer depth. For example, we can see that here in the positive phase, along Vancouver Island shelf, there is more concentration of chlorophyll and less during the negative phase. And as we have seen during the negative phase, there is a reduction in upwelling. So and upwelling bring to the surface nutrient that can sustain phytoplankton bloom during the season. So because there are this environmental difference, we were curious to see if there were also phytoplankton phenology difference during these two NPGO phases. So of course, this is a very preliminary results, and we look at we have a look just at the bloom peak time. So when basically the bloom reach its annual maximum, and here on the right we have our results. And if we compare, so we have the first figure here in the middle shows the bloom peak time during the negative phase, and then the other one shows the bloom peak time during the positive phase. And as we can see, during the positive phase, a large portion of the region shows late bloom peaks. However, again, I think that we need to look probably a different matrix and to have a complete, I mean, we need to go deep inside to this analysis to confirm or not this kind of results. And finally, we were also interested to see if there is a shift in the size structure of phytoplankton community. The table here on the left shows the phytoplankton size classes, micro, nano, and pico. And on the right, the figure shows basically 4 regions. And this time we didn't clusterize the time series like we did in the previous work, but this time these clusters are made just using the chlorophyll standard deviation because we were interested to create some region according to the annual concentration of chlorophyll A. So we have the oceanic area, which overall it's usually it's more oligotrophic. We have the southeast shelf, the green cluster here, and then we have the shelf and the shelf slope cluster, the light blue, which is again a transitional area and between the more productive coastal and the open ocean. So here we have the results. For reference, here we have our 4 cluster identified by different colors. And the first panel here shows the results for the 4 cluster for the negative phase. And the second panel on the right shows the results for the positive phase. So now let's have a look, for example, at the negative phase. So if we take in consideration, for example, the southeast shelf and the shelf, as we can see, there is a predominance of microplankton, and this is what we were expecting. But and then if we look at the oceanic region, there is a prevalence of in nanoplankton and picoplankton compared to microplankton, and this is also results that we were expecting. And finally, in the shelf slope, the difference are less evident because this is, of course, an area that it's a transition area between the two regime here. And however, if we compare the negative and the positive phase, the difference, there is not evident difference. So more work is needed eventually to find out if this difference are there or not. However, it is very, very interesting. It seems a very interesting results so far. So to conclude, some idea for the work that we want to do in the future. So we want for sure to improve the correlation analysis, also considering other variables such as upwelling index and nutrient concentration. I think that these two variables are, it is very useful to have a look at these two variables. And more significant highlight difference in bloom dynamics, employing eventually a specific algorithm to retrieve the matrix and then map phytoplankton phenology changes. And overall, we want to evaluate relationship between variables on a seasonal basis and possible among cluster, consistently discriminating between the NPGO phases. And thank you for your attention, and I stop here my presentation.
Thank you, Christian. Okay, I stop the registration and there is a space for questions. Either you raise your hand or you put in chat. Okay, thanks.